I'm very happy to have the opportunity to present this work. It's a really prime example of how we can utilize the AI immunology platform to really facilitate model development to meet medical needs. So just to rewind back a little bit to the previous talk where Thomas presented nicely how we have our Pioneer model to design personalized neoantigen vaccines. And he also presented that Pioneer is able to quantify the quality of the neoantigens. And he showed you this figure to the right, where you can see that the patients in our clinical trial, EVX-01, those patients that have a high neoantigen quality, they actually have a longer progression-free survival compared to those patients that have a low neoantigen quality predicted by Pioneer. But that of course also begs the question, so what about the patients who have a very few neoantigens of high quality? What can we do about these patients? Can we provide them with a personalized treatment that would be more efficacious than those that the neoantigens can provide? So you really need another antigen source to supplement the neoantigens in order to further optimize the personalized treatment here. And in that regard, I would like to introduce you to the endogenous retroviruses. For the rest of this talk, I will refer to these as ERVs. And the reason I'm introducing you to these would be that they are really promising targets as cancer antigens. What are these ERVs? They are ancient viruses which infected our ancestors thousands of years ago, and they have been lying in our DNA and they have been passed down through generations. They're very abundant in the genome, and normally they are not expressed in healthy tissue. But due to the dysregulation that happens in cancers, you suddenly have an expression of these ERVs, ERV-Antigens. So you really have the characteristic of a tumor-specific antigen here. There are a couple of examples in the literature that have described that ERVs can elicit specific T-cell responses in mice, and they can also protect the mice from a tumor challenge. Furthermore, there are measured in cancer patients, T-cell responses specific to ERVs have been measured. And finally, also in the laboratory, T cells that are specific to the ERV-Antigens can actually kill tumor cell lines. So all these characteristica really holds promise for the ERVs. So we then wanted to dive in and make some analyses in order to substantiate whether the ERVs would be a useful source of antigens in the personalized setting. The first thing we did was to do a very large-scale analysis across a lot of different cancer types on thousands of cancer patients. We analyzed their genomic profile and we counted up the number of neoantigens in the patient's tumors and also the number of ERVs being expressed in the patient's tumors. Interestingly, if you look here at the figure, then you can see that for each cancer type represented each by a dot, you can see the median number of neoantigens versus the median number of ERV-Antigens in the tumors for the patients. And what you can see is that they are not correlated. Furthermore, they're not correlated within each cancer type neither. And that's actually a good thing, because then if you have a patient who have very few neoantigens, then there could be the possibility to find a ERV sequence which would be useful for designing a personalized treatment. Yeah. The next thing we did in order to substantiate further whether ERVs as an antigen source would be efficacious, we looked into some cohorts of malignant melanoma patients treated with checkpoint inhibitor therapy. What we did was to analyze genomic profiles from genomic data from the baseline biopsies, and we then counted up the number of neoantigens in the patient's tumors and the number of ERVs being expressed in the patient's tumors. Then we split the patients into two groups. A group of patients with a lot of neoantigens, here labeled as high TMB. A group of patients with fewer neoantigens, here labeled as low TMB. If you focus on the plot in the center, then we further split the patients into groups based on the number of ERV-antigens being expressed in the patient's tumor. You can appreciate that these two curves fall on top of each other. So for these patients that have a lot of neoantigens, it doesn't matter whether you have a lot of ERV-antigens being expressed or few. However, on the contrary, if you focus on the plot to the right, then you can see the group of patients with few neoantigens. And if you then again split these patients into two groups based on the number of ERV-antigens, then you can basically see that those patients that have a lot of ERV-antigens being expressed in their tumor survive longer on the after checkpoint inhibitor therapy compared to those with very few ERVs being expressed. So this supports that ERVs could be a complementary source of antigens that could help the T cells to fight the cancer when there's very few quality neoantigens present in the tumor. So the next line of evidence we tried to investigate was, well, can we actually find these on the surface of tumor cells? So here we conducted some extra experiments in order to see which kind of MHC ligands do we have displayed on tumor cells. So we did that on two mouse tumors, one called the CD26 and one called the B16F10. And what we found was that ERVs are actually presented on MHCs, and we also found that our peptide MHC prediction tool, Evaxion, is able to predict these. So that's a really good sign that we can actually find these based on genomic data. So now we had a lot of lines of evidence supporting that we could use this in a personalized setting. So we started developing our model, ObsERV, utilizing our AI immunology platform. And we combined the different elements as such. But if we then look a little bit further down the hood, then I have created a little schematic for you here to really show the flow from patient to the design of a vaccine using ObsERV. So the first step is to collect a tumor biopsy from the patient, and that is to characterize the patient's HLA type and also identify those ERVs that are specifically expressed in the tumor. Then MHC ligands are predicted, and we rank the ERV-antigens, peptides within the ERV-antigens, based on the potency of the predicted MHC hotspots. You can then choose the top ranking peptides, and you can formulate that as a vaccine, for instance, as peptides or in a nucleotide format, which can then be delivered back into the patient. And the nice thing about this is that it fits into an established clinical workflow, and it really aims towards eliciting both the CD8 and CD4 T cell responses in order to achieve a sustained immune response, but also sustained effect. So now, having developed our ObsERV model, we wanted to test this out in an animal setting. So we designed a personalized ERV-based therapy for a mouse, and then we tried to vaccinate a group of mice with this designed therapy, and then we also vaccinated another group of mice with an empty plasmid, so that serves as a negative control. And then we saw then we challenged the mice with the CD26 tumor cell line and saw how well does the mice fare. So if you look at the plot to the right, then you can see how the tumor develops over time. It's a time course where you have the number of days after tumor inoculation along the first axis, and then you have the tumor volume on the y-axis. You can see that the negative control increases its volume exponentially, as expected, whereas the ObsERV designed vaccine actually completely prevents development of the tumor. The next thing we wanted to showcase was, well, Evaxion is a core component in ObsERV. So what if we try to swap this out with a different tool, a gold standard tool? So we tried to do that in order to really pinpoint the power of Evaxion. So we did that and also vaccinated mice, and this is shown as the red curve. And you can appreciate that that this vaccine does not completely prevent the establishment of the tumor. We then also looked into the T-cell responses, and you can see here on the most right figure that the specific T-cell responses towards the Evaxion-4 group actually is 3 times stronger as compared to that of the gold standard group. So it really underscores the importance of having a good MHC prediction tool. Finally, we also, more than 70 days later, tried to measure if these T-cell responses were really durable responses. So we measured again, and we saw that the T-cell responses were sustained. We also tried to do a secondary challenge on the Evaxion-4 group to see if they could still prevent establishment of the tumor, and was still completely prevented. So a very strong and sustained effect. Yeah, so now having established a proof of concept in the preclinic, we feel that we are ready to really test this out in a clinical setting. So that would basically be in a collaboration with a partner, the EVX-03 personalized trial. And this will be a trial where neoantigens and ERVs will be tested together. And it will be delivered using our in-house developed delivery technology, which is powered with a ABC targeting unit. So in this trial, you can imagine that you have some patients which have a lot of neoantigens. You can see that on the left figure all the way on the top. For these patients, they would have a lot of high-quality neoantigens and a lot there would be a lot of neoantigens within the vaccine design. If you go further down, then you have patients with fewer and fewer high-quality neoantigens, and here patients would then be given treatments comprising more and more ERV-derived peptides. So it's really a way to ensure that the most optimal personalized treatment is given to the patient. And this is how it fits into to the pipeline. So as I said, it would be the next personalized trial this would be tested in. So in summary, ERVs, they're a complementary antigen source to the neoantigens, and we have developed the ObsERV model for the design of ERV-based personalized cancer vaccines. And finally, also we are ready with the paperwork. We have been in communication with the medical agencies in order to see if this is viable, and we feel ready with the paperwork to really submit to initiate a clinical trial as soon as a suitable collaboration is established with a partner.